people coming in now. That's fantastic. So we've got a fantastic webinar for you lined up today. Before we get into the star attraction, let's just check everything is working okay. So if you can just pop a comment in the chat just to say that you can see the screen, you can hear me okay, that would be great. And I'll look for that, just check everything's working okay. And to say to you all today that this webinar is kind of brought to you by Carbotrace, and that is our fluorescent tracer molecules for labeling macromolecular components, specifically binding to repetitive glycosidic bonds. I'm guessing most of you in the webinar have kind of heard of this or interested, or even customers of Ebba Biotech, but that is what we will be focusing on today. And who better to tell you about this than our guest who will be presenting today. Bartosz Płachno is from the Jagiellonian University, professor at the Jagiellonian University, who was telling me before this that he loves kind of beautiful photos and beautiful research. And I can say that having been through some of your publications, Bartosz, I understand exactly what you mean, because they really are spectacular. I'm very, very much looking forward to hearing and seeing what you've got for us today. So without further ado, over to you, Bartosz.
Hello, everybody. It's a pleasure to say something about my research, and I would like to show you something about transfer cells in glands of carnivorous plants. At the beginning, I would like to describe shortly about the main subject. This is a transfer cell, and this transfer cell, the term was proposed by Professor Brian Gunning and Professor Jonas Stewart Pate from Queen's University, Belfast. However, this special cells was known in 19th century. This is special German term Übergangsschellen. So this is transition cells. This term was proposed by Fischer in 19th century for companion cells in the phloem. But what is amazing of these cells? These cells have a special development of the wall, cell wall. So you can see that cell wall forming like ingrowth here, and the membrane, plasma membrane, is also undulated. And this kind of the cells you can find in a generative reproduction structure, also in leaves, stem, roots. Also, this is one example that in algae, in one species of algae, these cells are also now known. And why the cells, they are in the tissue in the plants? Because these cells are important in the role in transport, especially that this undulating of membrane, in this membrane is also enriched in proteins. So every places that we have a big transport between apoplast and symplast in the plants, we have this specialized, this transfer cells. Okay, I would like to ask you, if you don't understand any subject and term, you can interrupt me every time and you can ask, okay? Okay, thank you.
Sounds good, sounds good. Yeah, you can chat, and I'll keep an eye on the chat as well so that you can pop questions and I'll ask them straight away.
And another question is, why carnivorous plants? Of course, I think that everybody knows carnivorous plants. This is very sophisticated group of the plants. And all this plant can attract, kill, and digest, and later absorb some nutrients from the bodies of animals, sometimes also the protists. And these plants produce special traps for catching of animals. And also in these traps, we can find the transfer cell. So we know from the book of Juniper, these transfer cells are in the glands of carnivorous plants. Especially also these plants, they have a different strategy for capturing the animals. And in traps, we can find different glandular structure which produce nectar, digestive enzymes, and also later they can absorb some nitrogen from the prey and also on another substances of the prey. So they are very good for our study. The cells of the glands are very active because we have here both secretion and later absorption. So this is very good research model for analyzing the cell wall, the membrane, and some organelles in the cells. Okay, so which method we use? We use the electron microscopy to see the ultrastructure of the cells. Sometimes we digest the cytoplasm to expose walls to see the micromorphology of the walls. We also used immunohistochemistry with confocal microscope to see which substances are in a cell wall and if any substances are changed during the development of the glands, or for example, during the secretion or absorption, if we have any differences. And of course, we all use also vital stains like DIOC or DEPI to see some organelles, to see some transport, and to compare our results with material from which we produced for ultrastructure. Why? Every technique produces some artifacts. So if we use different technique, we can compare the results and we have more holistic view what cells is, what the cells is active or not, and what is the problem, for example, of fixation of our technique, and we can avoid of some mistakes. And of course, it's very good opportunity to use some new technology like Carbotrace to compare our over results with your products. For immunohistochemistry, we use the primary antibodies, for example, anti-Arabinogalactans, anti-Pectins, and anti-Hemicellulose. So all substances which are commonly in a cell wall, we can use, we can check if they are in a cell walls or not. And also if some of the substances disappear or appeared. As I mentioned, we use ultrastructure. For ultrastructure, we use electron microscopy, and we use several types of the microscopes. So we use transmission electron microscopes. We use scanning electron microscopy for general morphology and also for some micromorphology of the cell. And you use also like a hybrid. This is scanning transmission electron microscopy. So this is a special scanning electron microscope that you can use the slides which are commonly viewed in transmission electron microscopy, but here you can also see ultrastructure of the cells. Okay, and this is example of the plants which we are studying. This is Dionea muscipula. This is a species from North America. This is very common in a cultivation. This is Drosera capensis from South Africa, and this is Nepenthes bilicalcarata, picture of the plants. So we have here 3 different types of the trap. Okay, and now Drosera. You can see that Drosera is not hungry because collect some fly, and here you can see the gland. And this small part, it consists of glandular cell, and the same part you have here, but it's a sectioned material. And we use here the reaction for polysaccharides to see the cell wall and to see some pectins in the cell wall. And also you can see here small like a dots. This is a transfer cell, and you can see wall ingrowth here. However, the resolution is not very high because this is only light microscopy. So we need more to see more details. And I would like to, in the rest of my presentation, show you more details using confocal and electron microscopes. Okay, we would like also to see micromorphology of cell wall in scanning electron microscope. But there are problems, because if you cut the cell, if you fix the cell, you have still cytoplasm and you don't see the cell wall. So we used very popular chemicals you can buy, I think, every shop in Europe, and you use this for destruction of the digestion of the cytoplasm to expose the cell wall. And here you can see examples. This is a part of the Dionea muscipula traps. This is digestive glands. So this is gland which produce digestive enzymes to digest the insects, and later the same glands, they absorbed nitrogen and other substances from the body of the prey. Okay, so now you can see two different techniques. This is photo from transmission electron microscope. You can see only a small part of the cell. This is cell wall. This is plasmodesmata, which are connect to cells. And this is wall ingrowth. This is cytoplasm. And this is mitochondria. One mitochondrium, second mitochondrium. So now the same cells, but after digestion of cytoplasm, this is scanning electron microscope. Here we used, and you can see the wall ingrowth. This here, that this wall ingrowth are forming like a network, like a labyrinth. Why? Because if the wall ingrowth forming network, labyrinth, also membrane forming, it's undulated. So this is for very, how to say, big transport between this cell and this cell. So this is for that we have symplastic transport, and from symplastic transport to apoplastic or in different way. Okay, another carnivorous plants, which I showed the picture of Nepenthes. This is gland from Nepenthes trap. And we cut the gland so we can see cells. And also we use digestion of the cytoplasm. You can see here, for example, this place. And you can see wall ingrowth here. So this is cell walls. We don't have cytoplasm because we digest this. And you can see that in some cells, this wall ingrowth forming the network. Okay. And now we go to Mexico, to the mountains. We see another carnivorous plant. This is Pingicula. I know that you have, I think, 3 species of Pingicula in your country. And in past, this Pingicula was used for special kind of the milk that people gave the leaf to the milk. And because this is carnivorous plant and produce some enzymes, so these enzymes made milk a special. So later this milk was used for different things. I don't know for this, but it was special for this. And I know this was in Scandinavia. And now you can see the prey, small fly, which are digested now. And here we have a very small glands. One gland is here, also digested. So we have a network of the wall ingrowth here. Okay, now we go to the Portugal. In Portugal, we can see Drosophilum lusitanicum. This is another carnivorous plant. In the past, this plant was used to catch the flies because it smells very nice like honey. But please don't try the substances of the leaf because it's completely not good. I did this in the past. So very smelly, beautiful smelly, but disgusting completely if you try this. This plant is very good in catching the flies. So a lot of flies, for example, if you have a fly problem, you can use this. And you can see that corpse of the animal here. This is, of course, scanning electron microscope, and you can see two different kinds of the glands. One is stalked glands, like here, and here you can see sessile glands. Maybe it's here is better. This is sessile glands here, and this is stalked glands. These glands have a different little structure, but different function, because these glands produce mucilage, and mucilage is very sticky. So if you would like to catch the animal, this mucilage glue the animal to the leaf. And after this, the small glands produce digestive enzymes. So we have two different glandular structure on the leaf, very specialized, one for glue animal, and this for digestion and later for absorption of nitrogen from the animal. Okay, everything, it's clear? Do you have any question?
It's clear. I was just wondering, how does the animal get to the digestive glands? Like, do they fall, or is there any way to kind of move them, or is it just natural that they will come into contact?
This is like, we say that it's a passive trap. So, for example, in Drosera, the leaf is active. So the leaf can move, and however, in Drosophilum, only the turgor of these cells are less, and later the animals has a contact with this digestive glands. But for example, in Drosera, we have an active leaf, active trap, but in Drosophilum not. And here we have an image of the stalked glands in scanning electron microscope. You can see the head here and the stalk. Inside the stalk, we have a vascular tissue which connects the glands with the vascular tissue of the leaf. And also very amazing was to find that this plant can take water from the fog. Because mostly carnivorous plants, they are growing in very wet places. Sometimes they are aquatic plants, but they like very wet. But this Drosophilum grows in very sunny but dry places in Portugal or Morocco. And researchers, they find that mostly the roots are dead. So in the beginning it was, oh my God, the roots are dead. It's very dry condition. That's why plants is still alive. But every morning they have a fog from the ocean, from the sea. And these stalked glands not only produce mucilage, but they also take the water from the air and transport to the plants. So they have a double role in a case the plants. Okay, so this is the stalked glands. And now it's the same glands, but it's sectioned with light microscope. You can see the head here and part of the stalk. And in case the head, you have different types of the cells. These two layers, this is glandular cells. So this is cells which produce mucilage, and this mucilage is produced outside later to glue the animal. And here we have another type of the cells. This is endodermal cells, Why endodermal? I don't remember, I don't know if you remember the lesson from the school about the roots of the plants. Do you remember the anatomy of the roots? Okay, I say very shortly. In the roots, we have a special tissue, it's endoderm. And in endoderm, you have a special cell which the cell walls are impregnated with the cutin or suberin, so hydrophobic substances. So all the transport should be via plasmodesmata, because the plant, you remember that cell wall of the plant, cell wall, this is a cellulose, pectins, and hemicellulose. So water can come everywhere. But if we have hydrophobic substances between the cellulosic, the water can't come. So all the transport should be via protoplast. So plant can control the transport. So in root we have this, and also in plant glands we have very, very similar, like here. You can see this is this, and this, and this. This is special cell wall which are with cutin or suberin. So transport between this cell and this cell should be here, not here, not here, but only using the plasmodesmata here. So from apoplast to the symplast, and later from the symplast to the apoplast. So this is about the control or transport in this way and from this way, okay? Also, please note that here we have a cuticle. This cuticle is very thick here, and in plants, cuticle is like a skin that it's protect plants from that the plants doesn't lose too much water. However, here the cuticle have a special structure. This is like open structure that plant can produce here substances outside, and also plant can absorb substances to inside the cell. So this cuticle has a different structure than cuticle here. Okay, and now the same gland, the head, and part of the stalk in confocal microscope. Of course, I show you only some example of our study. Here we would like to show Arabinogalactans proteins. Arabinogalactans proteins, this is very common proteins with some part of sugar parts, which are in most cell, but if you have a lot of them, this is a special cell, for example, for reproduction, or cell which are produce something, or for example, if there is any problem with bacteria, with pathogen, with fungi, so cell produce more arabinogalactans for some stress, for example. And here you can see that we have this green color. This is our arabinogalactans. This is GIM8. And you have some cells which are very rich in arabinogalactans. And you can see small dots here, some small dots, green dots. This is wall ingrowth, because this and here, this is a transfer cell in this Drosophilum gland. Okay. You can see more here. You can see that arabinogalactans are here, 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 here. For example, here. So this is transfer cell in this gland. And now another, you know that hemicelluloses are very common in some type of the cells, but there are difference between the different kind of the cells. And you can see, for example, that transfer cell are very rich in hemicelluloses, but for example, some stalk cell, they have none or they have only less. And here, for example, this is trachate. So this is xylan elements. And you can see that in some part of the cell wall, we don't have this hemicellulose. Only we have here, but not here. But the most they are very abundant. They are very common in the transfer cell. And this is galactan. You can see a different pattern of occurrence of galactan when we compare, for example, with hemicellulose. You can see, for example, in endodermal cell here, in some part in stalk, but for example, galactan is completely absent in the glandular cell here. So using different cell components, we can see that different type of the cells are in the glands. So they have a different function, and probably the cell wall, which is very flexible, the plant can, in every time they can should be modified. So also in case the occurrences of galactans, hemicellulose, pectins, arabinians, they is very flexible. For example, pectins. This is GIM7. This is methylated pectins. And you can see that these pectins are completely absent from the glandular cell here, but they are very common in the cell wall of the stalk here. But here, mostly not. Okay, and now we go to the digestive glands. This is one digestive gland. You can see some very similar characters like in a stalked glands, because you can see here glandular cell, 2 layers of glandular cells. This is our endodermal cell. So you can see modified cell wall with suberin and cutin here. And this is some basal cells which connect these glands with the mesophyll, and we can see also some tracheal elements of the xylan here. The same gland, different technology. This is scanning electron microscope photo. You can see the cells, glandular cells. Of course, we cut the gland. We later use some chemicals to digest the cytoplasm to show wall ingrowth here. For example, here. And you can see the wall ingrowth forming the network, like a labyrinth here. So, and now a different technique. This is glandular cell. I would like to show you the ultrastructure of the cell. This is STEM, so this is scanning transmission electron microscope. The tissue was specially prepared for this, so we use some chemicals that all the membranes, especially endoplasmic reticulum and nuclear envelope, are like a black. So the contrast is very high of this technique. To show you, this is cell wall, this is cuticle, this is cytoplasm, and in the cytoplasm you can see a lot of endoplasmic reticulum. You can see some mitochondria, some plastid, and this large structure. This is our nucleus in the cell. So this is this part of the cell, and I would like to show you also this bottom part of the cell that is a little different. This is also upper part of the cell. You can see membrane here and some undulation here. This is Golgi, some mitochondria, endoplasmic reticulum. And for example, for this we are not sure, is this chemical fixation, this structure, or this is natural. So maybe if we try to fix cells, but not using chemicals, but using the very low temperature cryofixation, we can say it's a true this undulation or not. Okay, and the same part of the cell, using the confocal microscope, here we use the diode to show the membrane, so especially endoplasmic reticulum, but also plastid and mitochondria, the green color, and the same cell, but using the calcafluor white to see the cellulosic cell wall. So the same picture and the same part of the cell which you saw here. Okay, now nucleus, endoplasmic reticulum, Golgi, mitochondria, and the same you can see nucleus, 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 and you can see like a radial pattern of the endoplasmic reticulum near the nucleus envelope. Yeah, okay. So this is whole glands in confocal microscope. This is also diode. This is a living cell. So of course, diode, it's a little toxic. So if we keep the material longer time, the cell will be dying. Yes, but in the first like a few minutes, you can see still living cell using this technique. So we can compare ultrastructure data with confocal data with living cell. So we can say, okay, so this is fixed killer cell and this is living, and we can see if we find some patterns of the endoplasmic reticulum, of distribution of mitochondria, of distribution of nucleus in a cell in a living stage and also in a fixed material. And this is bottom part of the cell. You can see cell wall ingrowth here, mitochondria, and plasmodesmata between two cells. And here you can see another technique. We use the special dye for membrane. So this is only, for example, diode, it's connected with the reticulum, with envelope of nucleus and plastid, but this dye only to cell membrane. And you can see diode and this membrane. So this is a membrane of the cell, and diode, this is reticulum and mitochondria and the plastid together in the one cell. And here wall ingrowth, you can see. So you can see that membrane, it's undulated. And please also note that we have endoplasmic reticulum, which is very, very close to the cell wall here. It's also wall ingrowth, cell wall ingrowth, and here we can see the cistern of the endoplasmic reticulum. This is a mitochondrium here. Also in this glands, in the cells, we have a very big plastids. I think that you normally know chloroplast because most plants are green and they make a photosynthesis, but a plant cell, they have a different kind of the plastids, and we say that some plastid, they are like a factories. So they produce a lot of metabolites. And if you check Drosophilum or Drosera dionea, you can find a lot of very active metabolics, which can be used, for example, for cancer cells, to destroy the cancer cells. They are antibacteria, antifungi. So, and we don't know why in the glands we have also this metabolics. Maybe this is because worse. If we carnivorous plants, they trap some prey, they digest. So plant doesn't want bacteria which could be take some nutrients for the same prey. So probably plants produce also this metabolic outside to destroy the bacteria and fungi. But of course, we can use these substances for our industry, for example, or for more medical reason, like for anti-cancer treatment. Okay, This was Drosophilum, but I would like to show you another model. This is Utricularia. I don't know if you are familiar with Utricularia. No? But you have 7 species of Utricularia in your country. But of course, Utricularia are not very known plants. This is in Europe. This is aquatic plants, but in the tropics we have also terrestrial, or for example, epiphytic plants. This is small green plants. All Utricularia produce traps. These traps are in water, and they are very sophisticated in case catching of the prey. One trap is like a blader, and we have a door. The trap is closed, and if any small animals touch the doors, the door it's opening, and the water with the animals goes very fastly to the inside the trap. This is one of the most fast plant organ. This is like milliseconds that this animal is taken with water to inside the trap. Of course, the door is closed, and now the plant can produce digestive enzymes, digest the animal, and later absorb the substances. And in this trap, we have a different glandular structure, very small epidermal origin. One of them, this is like, I say my student, this is like chromosome shaped. This is quadrifid. And also we have a very small glands outside. And why it's important? Because if the trap catch the animal and water, later the trap can be used again. But the problem is the water. So the water should be expelled from the trap. And some researchers, they propose that the water is it's absorbed by the glands inside the traps, later transported to the trap wall, and later it's produced outside using another glands. And this is the glands outside of the trap. In all Utricularia, they have very similar structure. This is epidermal origin. They have a terminal cell, 1 or 2. They have a pedestal cell, we can say endodermal cell, because this is walls which are impregnated with the cutin, and 1 large basal cell. And now, why it's interesting for us in case cell wall and elements of the cell wall? Because at the beginning, there are formation of the wall ingrowth here. So the terminal cell is a transfer cell. Also, this cell has a wall ingrowth, so this is also transfer cell. And during the trap development, in the terminal cell, we can see there are very thick cell wall. And if you can see here, this is a primary cell wall, and later we have a lot of secondary cell wall. And you can see in a light microscope, and you can see in transmission electron microscope, that the cell wall are different. Even the color, the structure of the cell wall different. So our idea is, okay, so let's go and check what is the difference in case the quality of the cell wall is another substances is here or here. So we did this. For example, Arabinogalactan proteins. This is from Confocal microscope picture. You can see that this is a primary cell wall, and this is secondary cell wall. And you can see that only signal, green signal is about Arabinogalactan proteins. This is a GIM8 antibody. And you can see that we have signal only here. So only the secondary cell wall possess Arabinogalactan proteins, but they are not in the primary cell wall. Of course, we wanted to check this in a high resolution technique. So we use a transmission electron microscope, and you can see here the picture from transmission electron microscope. This is this part. So this is this part, and you can see primary cell wall is here and here, and this is wall ingress, and this is secondary cell wall. And of course, we use antibody with gold particles. So you can see here, 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 gold particles here. This is a higher magnification here, but mostly not gold particles in the primary cell wall, only in the secondary cell wall. So this is very nice picture that we have a different cell walls, primary, secondary, and there are difference in case occurrence, for example, Arabinogalactans proteins in this particular in GIM8. Any questions?
So I just had one thought about when the water is kind of coming out and the digestion process.
Yeah.
Like what happens first? Because if the water is inside when they need to digest, then that would require probably a lot more digestive substances. So, but then of course it may be more complicated to get the water out first. Do you have an idea of the timing of that?
They need some few hours to be ready for catching to another animal. I think the first is digestion, and later it's removing the water. But of course, a little water still exists in a trap. I didn't mention it, but there are some algae, for example, and this algae, for example, can living in the trap, so they are not digested. And also, if you compare very young trap and very old trap, so the young trap, it's more carnivorous, so they are digest more. But if you have older and older trap, you have a lot of some debris, some living animals inside the trap. So it's like a microscopes inside. So plants are more like a mutualistic in case, but not like carnivores. But in the fresh trap, young trap, it's more carnivorous, but in older, it's a more mutualistic, some symbiosis with some bacteria and some small animals. And this is, I think, for you, it could be interesting because this is Hemicellulose. This is LM25. And you can see this is terminal cell, this is pedestal cell, basal cell. So this is the same gland when we saw. And you can see that we have here a lot of hemicellulose here in the secondary cell wall. You can see layer and layer that it was the first this, later this, and this is, so it was build. And Carbotrace. I find on your page that you informed that Carbotrace is good for cellulose and also for hemicellulose. So you can compare this picture with this picture. So I think ideal situation that here we have an agglomeration of hemicellulose, especially LM25, but also LM50. I don't show this picture, but we have also this result. And now the Carbotrace here. And of course, we use also calcafluoride, as you can see, very similar situation to Carbotrace. Okay, but the story is still going. Now I would like to show, because it's the same trap, this is Utricularia, but we can now see these glands. And I think that everybody in this room know Charles Darwin. Yes. And I don't know if you know that Charles Darwin was author of a book about carnivorous plants. and he wrote letter that more amazing for him is Drosera and how Drosera catching the fly than origin the species. So, good for us that Charles Darwin, not use his all time for carnivorous plant, but he wrote only one book about carnivorous plants, not more. And he studied Utricularia, and he studied the glands, and he proposed the term quadrifids, because you can see here that we have a four cells here. Of course, in this time it was not electron microscope, so he didn't know proper structure. Later, Professor Juniper, using electron microscope, showed that this gland, they are very complicated, especially the terminal cell, because we have a basal cell, pedestal cell, this endodermal cell, so we have some part of the cell wall which are impregnated with chitin, and also we have a special terminal cell, and each terminal cell have an arm here, and have a stalk. And the stalk of the cells are united together. So you have four cells. Of course, here we can see only two, but you can see we have four cells which are united with stalks, and only three, they are arms. So very small origin, epidermal origin, but very complicated cells. And this cell is a transfer cell, and this cell is also transfer cell. So this is also ideal situation to study specialization of different part of the cell wall in different part of this cell. Okay, so this is section of the quadrifid. You can see arm, the stalk, and the pedestal cell and basal cell, and this part you can see here, and you can see here. So I told you that pedestal cell is a transfer cell, and you can see here, this is cell wall. This is wall ingrowth. And here is mitochondrium. So mostly cell, it's full of the wall ingrowth. So this is very, very active cells with a lot of numbers of mitochondria. And this cell wall, outer cell wall, this is this part, it's impregnated with chitin. This is antibody GIM5, so this is a pectins, non-esterificated pectins. And you can see that we have a green color here, so these pectins are here, no in wall ingrowth, so here not, and also not here. So in this part, we don't have this pectin, but the question is now very important. Or we don't have this pectin here, or impregnation with some hydrophobic substances blocked antibodies. So antibodies can't come to the pectins, okay? This is another pectins. This is LM19s. You can see now this cartoon that you can, because unfortunately this is very not nice material for cutting. So we have like a fragments of the cell. So this is, we draw something that you can see. For example, this part is this part, this part is this part, this part is this part. So you can see distribution of this pectin. So you can see, for example, that pectin is in this cell's walls, but no in wall ingrowth, but for example, in cell wall of the arm. Another pectins. Here you can see this pectin occur in cell wall ingrowth, like here. Okay, in this part, but not in this part. You can see black here. GIM7, This is estrophylated pectins, and you can see another distribution completely different to this. Please note this, and now see this. So we can see the difference between, even in case pectins, you can see the difference. And now hemicellulose, we have, I'll show you on the two, this is a Xylan and Xyloglucan, and you can see a difference. For example, Xylan is absent here in wall ingrowth, and Xyloglucan, you can see wall ingrowth here, that it occurs here. You can see, please see this and see this. So finally, Carbotrace. One thing which I think this could be important for you here, that we find things like here, and when we see the same slide in the light microscope, we find that this is bacteria. So Carbotrace also could, you can see some bacteria wall. So this could be for you interesting. And this is interesting. This is a part which is impregnated with cutin. So this is part of the cell wall which is impregnated with hydrophobic substances. And we have a, not problem, we find that Carbotrace doesn't work here. But we know that this is a cellulosic part, yes? Because this is cell wall, and cellulosic has to be here, but probably with hydrophobic substances like cutin or suberin blocked Carbotrace to come to the cellulosic. So I think this is a nice idea about developing the procedure and to see with over Carbotraces, because we use only one type, to see if we have a similar results or not. Okay, so what we find that cell wall ingrowths are rich in Arabinogalactans, mostly in all carnivorous plants which we study. We found also that there are different types of cells in glands with different cell wall composition, in case pectins, hemicellulose, and Arabinogalactans. And unfortunately, we only checked in Venus flytrap in Dionea stipula that we find that wall composition can change after the feeding. But still, we need to develop this because it was very time-consuming research. So we plan to do this with over carnivorous plants, but of course we need money and we need the time for this. Okay, and also here I would like to show that we have cell wall microdomains, and after checking different components, we can see different microdomains in case one cell or in case gland, for example, like here. So wall ingrowths are different the microdomain. This impregnated cell wall is completely different microdomain, and for example, the arm of the glands is different microdomain, and also wall ingrowths here in terminal cell are different domains in case lacking or occurring some cell component. Okay, and in case Carbotrace, this is very nice that we find coincidence with detection of hemicellulose. However, we have this negative results in case staining with Carbotrace in cutin impregnated cell walls. I don't know, because only a few paper was published in case plant anatomy, in case Carbotrace. So I couldn't find information if somebody finds similar results. But I think this is very good to check in another plant material, and for example, some roots, to see if it's suberin also will be the same like with cutin impregnated cell wall. And of course, with the different kind of the Carbotrace, if it's the same results or not. And our smart team, okay, I'm here, but we have my colleague Piotr from another university, Małgosia from Gdańsk University, and also Irene from University of Vienna. So if you see, every people from another universities, so it's like a virtual team. And we published some papers in some journals about this on some examples that we use different antibodies to show the cell wall carnivorous plants, but also, of course, see some ultrastructure and the activity of the glands and activity of the cell of the carnivorous plants. Okay, so thank you very much for your attention.